Hi, my name is Jay McCurdy. I'm an assistant professor at Mississippi State University and a co-principal investigator working alongside a nationwide group of faculty seeking to understand the ecology and biology of the troublesome turfgrass weed Poa annua, or annual bluegrass. Welcome to Module 3, which covers herbicide sites of action. If you haven't already done so, please make sure to watch the previous modules for a more in-depth understanding of annual bluegrass and the problem of herbicide resistance. In this module, we'll discuss rotating herbicide sites of action in order to preserve the use of these chemistries. Annual bluegrass has historically been an important weed of many, if not most, commodity and specialty crops. The extensive reliance upon herbicides as the primary means of control has led to almost overwhelming presence of herbicide resistance. There are very few commonly utilized herbicides that annual bluegrass has not evolved resistance to, albeit often in isolated or unique populations. The worrying trend is that for some turf scenarios, we no longer have effective chemical means of controlling annual bluegrass. The sites of action listed in this table represent the currently reported cases of annual bluegrass resistance to various herbicides. The center column contains the WSSA shorthand numbers representing each of these sites. We'll learn more about these later, but they are meant to help practitioners to quickly and easily distinguish between different sites of action. The active ingredients reported in the column on the right represent relevant active ingredients within the turfgrass industry. Many of us will recognize common active ingredients such as simazine and atrazine or glyphosate, which are standard means of chemically controlling annual bluegrass. You may also be familiar with cases of suspected herbicide resistance to the likes of trifloxysulfuron, the active ingredient in Monument, or 4-amsulfuron, the active ingredient in Revolver. These acetolactate synthase inhibitors are frequently used for control of annual bluegrass in warm season scenarios. Herbicide-resistant annual bluegrass is an increasingly difficult problem for professional turfgrass managers. Controlling it requires an integrated approach, including appropriate grass species or variety selection for the intended use, performing cultural practices that favor the desired grass, and preventative control strategies such as quarantining suspected resistant populations and using clean sod and seed sources. When using herbicides, use full labeled rates at proper timings. Use multiple sites of action within overlapping spectrums of weed control in rotation between years, in sequence within seasons, and/or in mixture at the same time. Finally, scouting for escaped weeds after a herbicide application is often necessary in order to avoid weeds going to seed or proliferating vegetatively. This means scouting and eradicating escapees after treatment. The slides that follow discuss herbicides by their overarching symptomology. We then discuss site of action in greater detail and offer examples of specific herbicides used in the turf market to control annual bluegrass. There are several herbicides commonly used to control annual bluegrass that work by disrupting the synthesis of amino acids. Amino acids are organic compounds containing an amine group and a carboxyl group. Each amino acid also has a unique R group or side chain. Amino acids form bonds to create proteins and are also responsible for numerous other physiological roles within plants. Inhibition of their synthesis leads to decreased production of proteins and nucleic acids like DNA and RNA, as well as disruption of other plant functions. Group 2 herbicides are the acetolactate synthase, or ALS, inhibitors. The ALS enzyme functions within a key step in the synthesis of branch-chain amino acids, such as isoleucine, leucine, and valine. ALS inhibitors are notorious for their selectivity, both for selective weed control and for crop safety across various turfgrass and commodity ag species. The sulfonylurea family of herbicides include Katana, flazosulfuron, Revolver, 4-amsulfuron, Monument, trifloxysulfuron, Certainty, sulfasulfuron, and the active ingredient rimsulfuron, which is both negate and transit, or other generic products such as QualiPro product pictured here. With slight exceptions, none of these herbicides are labeled for use in cool season scenarios. It is also worth noting that these herbicides have a range of safety on warm season grasses, but that all are labeled on Bermuda grass. The pyrimidinal thiobenzoate family includes only one ALS-inhibiting turf herbicide, which is bispirabac sodium, the active ingredient in Velocity. Velocity, unlike the sulfonylureas family, is labeled in various cool and warm season grass scenarios. ALS-inhibiting herbicide resistance has been confirmed in at least 160 different weed species, including annual bluegrass. Resistance is attributed to the widespread use of these once very effective herbicides for post-emergence annual bluegrass control. Several mutations within the gene encoding for the ALS enzyme can result in herbicide resistance. Although herbicide resistance is increasingly common for this group of herbicides, it has not relegated them to the past. These herbicides, especially when combined with other sites of action, still play an important role in the control of annual bluegrass. Glyphosate is unique in that it is the only commercially marketed herbicide within Group 9. Like the ALS inhibitors mentioned previously, it too interrupts amino acid synthesis. However, glyphosate inhibits 5-enol pyruvyl shikimate 3 phosphate synthase, known as the EPSP enzyme. This blocks the production of aromatic amino acids, tryptophan, tyrosine, and phenylalanine. Glyphosate is one of the most widely used herbicides within row crop commodity agriculture. The advent of glyphosate-tolerant or Roundup-ready crops played a large role in its prominence. However, a problem with the widespread use has been selection for glyphosate-resistant weeds. At least 43 weed species have been confirmed resistant to glyphosate, including annual bluegrass. Due to the non-selective nature of glyphosate, it is most commonly used to control winter annuals, such as annual bluegrass and broadleaf weeds, in dormant Bermuda grass and zoysia grass. This has somewhat confined its use to the transition zone of the United States. Hypothetically, this zone of use may have more populations with resistance to glyphosate. However, glyphosate is also commonly used at sublethal rates for growth suppression and chemical mowing of transportation right-of-way. The sublethal rates used only serve to expand and spread herbicide-resistant populations. Group 3 encompasses a number of different herbicides with pre-emergence and, to some extent, post-emergence weed control. These herbicides interfere with cell division by preventing microtubule formation. Microtubules are the building blocks of spindle fibers that allow for chromosome pairs to be pulled apart during mitosis. Hence, we often call these mitosis or mitotic inhibiting herbicides. During the early stages of seedling germination and growth, an embryonic root or radicle emerges and quickly seeks to move downward into the soil where it stabilizes and supports the emerging shoots. When that radicle comes into contact with a soil-applied herbicide, it can no longer grow correctly. But these herbicides in many instances also negatively impact the desired turf species by causing malformed roots and rhizomes or decreased pegging of above-ground stolons. These herbicides are sometimes referred to as root pruning for this reason. However, when used at correct rates, the risk of turf decline is relatively low. There are many herbicides included within this group. The dinitroaniline herbicides, Barricade, prodiamine, and Pendulum, pendimethalin, as well as the somewhat older herbicides, oryzolin and trifluralin, are predominantly used as true pre-emergence herbicides for control of annual grasses and broad-leaved weeds. These herbicides are commonly orange to yellow in color and are sometimes referred to as such. The pyridine herbicide Dimension, dithiopyr, has both pre-emergence and some slight post-emergence control of annual grasses and broadleaves. Similarly, the benzamide herbicide Curb, Pronomide, controls annual bluegrass both pre- and post-emergence. For both dithiopyr and pronomide, post-emergence control is limited. There are at least 12 confirmed species that are resistant to microtubule inhibiting herbicides, including annual bluegrass. There are many photosynthesis inhibiting herbicides. In fact, most herbicides in some manner reduce photochemical efficiency, the process by which plants process light and turn it into usable forms of energy. Group 5 herbicides include those that inhibit photosystem 2. All of them inhibit electron flow during the light reactions of photosynthesis. This stops carbon dioxide fixation and the production of adenosine triphosphate, ATP, a compound that provides energy for most physiological processes within plants. Membranes are also degraded during this process due to the overabundance of energy absorbed by light-harvesting chlorophyll and carotenoids within the plant. Ultimately, this leads to electron leakage and inability for membranes to function properly. The triazine family includes two very common pre- and post-emergence herbicides, Atrex, atrazine, and Princep, simazine. Both are labeled for use in warm season scenarios, predominantly for control of annual grasses, such as annual bluegrass, as well as for control of broad-leaved weeds. The triazinone family includes Syncor, metribuzin, which shares many of the same uses and weeds controlled as atrazine and simazine. And the triazolinone family includes Exonerate, amicarbazone, which is one of the few PS2 inhibiting herbicides labeled for use in cool season scenarios, including creeping bentgrass, but annual bluegrass control is sometimes inconsistent. These PS2 inhibiting herbicides are all similar in that they are readily absorbed through roots from soil applications and are translocated to shoots. They have varying degrees of absorption into foliage and subsequent movement throughout the plant. Although not included in this discussion, there are at least two different groups of PS2 inhibitors, including herbicides such as bentazone, bromoxynil, siduron, and diuron. Names many of us have heard before, but have limited relevance to control of annual bluegrass. The exception is diuron, which has been commonly used in seed production. Note that there are annual bluegrass populations resistant to diuron in the Pacific Northwest seed production fields of the United States. There are at least 74 species confirmed resistant to Group 5 photosystem 2 inhibiting herbicides. In fact, resistance is fairly common across many annual bluegrass populations. In the pictures shown here, one can easily see that this population of annual bluegrass is poorly controlled by atrazine, while it is well controlled by other common pre-emergence herbicides. It is important to distinguish that in many instances, poor activation, uptake, or residual of a herbicide may impact efficacy. That was not the case for this population. It is confirmed resistant. Group 10 herbicides include just one common turf active ingredient, that of phenale, glufosinate ammonium. Inhibition of glutamine synthase, which converts glutamate and ammonia to glutamine, leads to buildup of ammonia, which destroys cells and inhibits key light harvesting processes within the plant. There are only 4 species confirmed resistant to this herbicide, and annual bluegrass is not yet one of them. Phenale is a non-selective herbicide confined to use within the transition zone where warm season species experience dormancy, particularly Bermuda grass and zoysia grass. Group 14 herbicides include the protoporphyrinogen oxidase, PPO or Protox, inhibitors. There are an array of PPO inhibitors within the broader ag and turf markets. The ones with utility for annual bluegrass control and maintained turf include SureGuard, flumioxazin, and Ronstar, oxadiazone. The N-phenolphthalimide, flumioxazin, is used predominantly for post-emergence burn down of annual bluegrass in dormant turf. Its rapid desiccation of foliage limits its translocation to the roots. But when soil applied, it does have some residual control and translocation to foliage of immature plants. The oxadiazole, oxadiazone, is used more broadly as a pre-emergence herbicide in turf than flumioxazin. When applied to the soil, it is readily absorbed by emerging seedling foliage. When applied to foliage, it causes rapid bronzing and desiccation of weeds and turf alike. Key to the use of these PPO inhibitors is the delivery method. Both flumioxazin and oxadiazone are available on granular carriers for soil application and as powders or liquids for broadcast liquid applications. Using granular application methods reduces the risk of foliar injury of non-dormant turf and other plant material. The distribution of granular material, both in terms of size and prills per unit area, may impact efficacy due to poor coverage or lack of uniformity in coverage. There are at least 13 species reported to be resistant to PPO inhibitors. Annual bluegrass resistance has not been reported, but is suspected in certain populations. Furthermore, in the last few years, goosegrass resistance to Ronstar has become a topic of concern. Liquid broadcast application of Ronstar is generally limited to use in dormant turf, whereas granular Ronstar can be used in non-dormant warm season and cool season grasses. Both flumioxazin and oxadiazone have various labels for use in landscape ornamentals, but may be restricted for use in home lawns. Group 22 herbicides interrupt photosystem 1 and are more generally called cell membrane disruptors. They act by diverting electrons in the dark reaction of photosynthesis and by becoming energized molecules known as herbicide radicals. These radicals lead to destruction of membranes within the plant. Control and desiccation are rapid. Thus, these herbicides are not well translocated. These herbicides are extensively used as burn-down herbicides in row crop and commodity agriculture. The bipyridinium family includes the non-selective herbicide Reward diquat. Reward is used predominantly in dormant warm season turf scenarios for rapid control of winter annual broadleaves and grasses. There are at least 32 species confirmed resistant to this site of action, including annual bluegrass. Group 15 herbicides are sometimes discussed more broadly with other shoot growth inhibitors. The very long-chain fatty acid synthesis inhibitors are typically applied as pre-emergence or soil residual herbicides and lead to aborted seedling or embryonic shoot formation and inability to form fundamental plant structures, such as leaf cuticles. They do not necessarily inhibit seed germination, but instead affect structural formation of embryonic plants, whether emerging from seed or growing from vegetative structures such as rhizomes or tubers of nutsedge. The chloroacetamides Tower, dimethenamide, and pennant magnum, metolachlor, are labeled for control of annual grasses, broadleaves, and sedges. However, tank mixing with either simazine or atrazine is a common practice to enhance pre-emergence activity on annual bluegrass and broadleaf weed species. There are at least 5 species confirmed resistant to this site of action. However, annual bluegrass is not yet amongst them. Cellulose biosynthesis inhibition is a broad term used more appropriately to discuss the mode of action rather than specific site of action of a herbicide. In fact, there are several groups of herbicides within the larger umbrella term. Group 29 is amongst the newest sites of action discovered by herbicide physiologists. The alkylazine herbicide Spectacle, indaziflam, inhibits cell wall biosynthesis by interrupting cellulose synthesis. Cellulose plays a critical role in cell wall strength and plant growth. Although the specific site of action is not well characterized, Group 29 is clearly different from Group 20 and 21 cellulose biosynthesis inhibitors, which are not discussed. To date, indaziflam is the only developed herbicide amongst Group 29. When root absorbed, it leads to clubbed root formation and stunted foliar growth. Spectacle is also active when applied to foliage, though it may lack efficacy without root absorption. To date, no reports of herbicide resistance to this herbicide have been confirmed. However, there are instances where efficacy is compromised due to environmental conditions. This herbicide is labeled for use only within warm season scenarios. Group 27 herbicides inhibit the enzyme 4-hydroxyphenyl pyruvate dioxygenase, HPPD. The triketone herbicide tenacity, mesotrione, is labeled for pre-emergent suppression of annual bluegrass. There are at least 2 species that are confirmed resistant to HPPD inhibitors. Due to limited expectations for control, its use for annual bluegrass suppression is limited to areas where other herbicides are lacking, such as cool season turf scenarios. Group 30 herbicides are quite new and lesser understood in the realm of maintained turf. The herbicide methiazolin is not yet registered in the United States, but has been shown useful during the pre-registration research period. This herbicide inhibits tyrosine aminotransferase, which is a key step in the production of cofactors for normal plant function, including plastoquinone. Although not yet registered, methiazolin will be a unique chemistry for selective pre- and post-emergence grass control. The precise sites of action of the fatty acid and lipid synthesis inhibitors are not completely understood. Group 8 includes the phosphorodithioate, benzimec or betasan, benzoylide, which is used for pre-emergence control of various annual grasses, including annual bluegrass. It is less commonly applied as a standalone active ingredient by professional turf managers. Many turf managers will know of Anderson's goose and crab, which contains both benzoylide and the PPO inhibitor oxidizone. Benzimec and Anderson's goose and crab are some of the few pre-emergence herbicides labeled for use on golf course putting greens. The benzofuran prograss, ethofumicate, is labeled for both pre- and post-emergence control of annual bluegrass. Prograss is labeled for use on a number of cool season turfgrass species and for use in St. Augustine grass for Bermuda grass suppression. Along those same lines, it cannot be used on Bermuda grass until dormancy without risk of injury and delayed spring greenup. There has been at least one reported case of annual bluegrass resistance to ethofumicate in the state of Oregon. In addition to commonly classified herbicides, other chemical means of control also exist. There are many plant growth regulators, or retardants, that are commonly used to suppress foliar growth and seed head formation. The triazole trimet, paclobutrazol, is classified as a fungicide, but also is a known antagonist of the plant hormone gibberellin. By inhibiting gibberellin biosynthesis, it reduces internode growth and seed head formation. The phosphonic acid proxy, ethofon, is labeled for use in commercial turf for seed head suppression of annual bluegrass and several broadleaved weeds. Its liberation of ethylene interferes with the plant growth process. It is also commonly used to accelerate fruit ripening. The acyl cyclohexane dione primo trinexapac ethyl is labeled for seed head suppression and growth regulation of annual bluegrass. It can be tank mixed with trimet and proxy for improved seed head suppression. None of these plant growth regulators truly control annual bluegrass, but when applied in a program on high-end turf scenarios, they do manage or reduce populations. Planning herbicide programs is an important strategy to mitigate risks of resistance. In the short term, an approach based upon the correct best management practices will result in better control. In the long term, better control will decrease the amount of annual bluegrass seed in the soil seed bank and will decrease the chances of resistance in the future. Fundamentals of herbicide programming include an emphasis upon pre-emergence herbicides to prevent overwhelming plant density. This approach limits population size and thus the likelihood of a resistant plant gaining a foothold is diminished. With any herbicide application, there will be uncontrolled plants. It is fundamental that practitioners follow up with post-emergence or non-chemical control strategies to minimize escapees and their reproduction. Proper herbicide selection is fundamental to chemical control of annual bluegrass. If it doesn't result in thorough control, then it is more likely to have selection pressure for full-blown resistance to the chosen herbicide. There are inherent risks associated with various herbicides, including risk of injury on desired turf species. Always make sure that you have read and understood label recommendations. This table is an example of a poorly planned rotation between herbicides. That rotation will eventually fail because of reliance upon similar sites of action. So what's wrong with this POA program? For starters, each year relies upon the pre-emergence mitotic inhibiting herbicides pendimethalin, dithiopyr, and prodiamine, all of which are the same site of action. You can see that they share the WSSA group number 3. The post-emergence applications, the ones applied after annual bluegrass has emerged, are again all the same group number associated with herbicides that inhibit the enzyme acetolactate synthase, group 2. Take note, however, that this program could not be or would not be suitable in cool season grass scenarios. Here's an example of a program that correctly rotates sites of action both within and between years. In year 1, the photosystem 2 inhibitor simazine is applied and followed up with the ALS inhibitor 4-amsulfuron. In year 2, the mitotic inhibiting herbicide prodiamine is applied and followed up with the non-selective glutamine synthase inhibitor glufosinate. In year 3, the cellulose biosynthesis inhibitor indaziflam is followed up with the photosystem 2 inhibitor atrazine. And in year 4, the PPO inhibitor oxidazone is applied and followed up with the mitotic inhibiting herbicide pronamide. Again, take note, this is a program that would not be suitable in cool season grass scenarios. An even better example of herbicide programs for warm season scenarios might include multiple sites of action within applications. For instance, here you see the mitotic inhibitor prodiamine applied in combination with the ALS inhibitor trifloxysulfuron. Then in the same year, photosystem 2 inhibitor simazine is applied as a follow-up. In year 2, a different approach involves the cellulose biosynthesis inhibitor indaziflam, followed by 2 different sites of action, the ALS inhibitor 4-amsulfuron and the mitotic inhibitor pronamide. Again, this approach is less feasible in cool season scenarios due to the extremely limited number of herbicides effective against annual bluegrass and tolerated by cool season species. Effective herbicide resistance management requires a balance of chemical and non-chemical tactics. Best management practices include decreased reliance upon chemical weed control and increased diversity of traditional cultural practices, such as proper turf species selection for the scenario or environment at hand. The emphasis should be on providing a dense and competitive turf sward. A holistic approach of managing soil and plant health is required in order to do so. When herbicides are used, we must diversify our sites of action between years and within season. And we must also emphasize pre-emergence followed by post-emergence means of control, which effectively reduces the amount of annual bluegrass exposed to post-emergence strategies. Finally, these authors, as well as team members within their labs, are to be given credit for the development and review of this module. Financial support for this work was provided by the United States Department of Agriculture Specialty Crop Research Grant. Our thanks are extended to the Weed Science Society of America and Herbicide Resistance Action Committee for allowing us to use training material posted on their website as the starting point for these lessons.